Welcome to this first ever pitch showcase of the NE's call to action for engineering solutions to mitigate the spread and impact of COVID-19 pandemic. I am John Anderson, president of the National Academy of Engineering and a chemical engineer by education and practice. With hundreds of thousands dead and economies collapsing around the world, the engineering profession must proactively respond to this urgent crisis. This is why the NEE was created, to bring creative engineering expertise to bear in order to address vital needs of society. This call to action and unprecedented engineering initiative was sparked by a mobilization of enthusiastic students and alumni of the NEE Grand Challenges Scholars Program, who basically said, this moment is what we've trained for. At the same time, alumni of the elite NEE Frontiers of Engineering program announced they would like to be involved in brainstorming how engineering could make tangible contributions to addressing the impact of the pandemic. Our programs office, led by Randy Atkins, Janet Hunsinger, and Guru Madhavan, suggested we combine these two groups, the young and middle-aged professionals, to gain broader perspective of how engineering can contribute. Then some of our NEE members, we'll call them the senior ones out of respect, suggested joining forces with the call to action to add their experience and professional networks to this initiative. So here we are, a first of its kind virtual incubator for developing engineering solutions to a variety of COVID-19 related problems. And I think as important as that, engaging three generations of engineers. The younger engineers learn from the older engineers, and the older engineers learn from the younger engineers. This is an intricate, multi-level process which includes the NSF I-Corps Business Development Training. More than 700 people have joined the Call to Action platform. You can learn more about this call to action on the NAE website, which is at nae.edu. And if you are not participating already, you might decide to join us. Today, we will showcase 5 concepts that have gone through the process from idea to a place where they may be ready to launch. There will be more of these events in the weeks to come. We hope the NAE call to action will prove to be a platform to spark engineering innovation for challenges beyond this pandemic and inspire all engineers, all generations of engineers. I wish to thank the many people who have made this initiative possible, including those in the NSF Innovation Node Los Angeles who have trained our teams, as well as the support of NSF I-Corps program. Sponsors who have donated resources to make this happen. Dr. Yanis Yortzis, Dean of the USC Viterbi School of Engineering. Andrew Mong and Rachel Lau, alumni of the Grand Challenges Scholars Program, who co-founded this initiative with us. Frontiers of Engineering alumni on the Technical Screening Committee. The NAE members you will see today on our Expert Review Committee, including its chair, Paul Citron, and the many volunteers and participants who have made the call to action such an exciting and important initiative. I now turn things over to Dr. Alice Liu, who will moderate today's event. Alice is with the Innovation Node in Los Angeles. Over to you, Alice.
Thank you, John. This is truly an exciting, important initiative. I'm proud to moderate the pitch showcase portion of the program today. Before we begin today's pitch showcase, let me lay down some ground rules. The format for today. Each team will be giving a 5-minute pitch followed by 8 minutes of Q&A with our Expert Review Committee. We refer to them as the ERC. They are not judges, as this is not a competition. Our goal here today is to help advance these ideas and accelerate their trajectory to reality. This includes your participation, our audience viewing from home. If you have comments or thoughts or think of ways to help as you're listening, we encourage you to tweet and use hashtag NAECOVID19. You can also send your comments to engcovid19@nae.edu. We are honored to have a very distinguished panel of reviewers on the ERC today. In total, we have 12 reviewers who have been divided across the Q&A sessions. I will introduce each of our members of the ERC panel at the start of each Q&A. Now, without further ado, I'd like to welcome up our first team. Team, wearable sensors for remote monitoring of COVID-19 patients. Shu, you have 5 minutes. Are you ready?
Yes, we are ready. Hello, everyone. We are Team Coverty from UC San Diego, and we are bringing a remote monitoring Band-Aid for primary care doctors for COVID-19 patient triage. Next, please. Next slide. The COVID-19 pandemic has caused turmoil all over the world. In the US alone, there are now over 2.3 million patients who tested positive, and for 80% of them, symptoms remain mild to moderate. But for about 19% of cases, patients start out with mild symptoms, but quickly deteriorate in the second week. Mild to moderate COVID patients are asked to self-isolate and contact a doctor if certain warning signs appear, because early detection of deterioration helps provide timely intervention to save the patient from a downhill path, or at least make sure there's a ventilator available if needed. Next, please. How rapid can COVID-19 progress? I want to share with you the journey of an individual patient. On March 27, 2020, the patient tested positive for COVID-19. At the time, his symptoms were only mild and he remained in self-isolation at home and kept working for the whole time, actually thinking things were not as serious. On April 5th, his symptoms worsened and he was admitted to a hospital. And on the 6th, he went into ICU. It took him only 10 days from mild symptoms to ICU. Next, please. The patient we are talking about is Boris Johnson, the UK Prime Minister. Now, if a well-informed political figure under the advice of his healthcare team cannot have a correct estimation of how serious his condition was, how well can a general patient do it? Next slide. In a pandemic, how can doctors make sure the patients will visit when necessary and only when necessary? Patient triage is increasingly relying on remote consultations, but the challenge is that doctors don't have access to the patient vitals because there are no validated tools to assess key symptoms remotely. For example, a key differentiating factor for severe cases is breathlessness. But with everyone's baseline different, how do you quantify that? And what if symptoms worsen during sleep? Next, please. At Coverly, our team is building a platform for remote monitoring of COVID patients to help doctors make informed decisions. The proposed device concept is a soft patch that sticks onto the skin like a Band-Aid that can measure key vitals such as respiratory rate, heart rate, temperature, oxygen saturation, and blood pressure in continuous manner, and send automatic alerts to doctors when the vitals drop out of desired range. Next, please. The concept is based on our patented technology published in Nature Biomedical Engineering, which uses ultrasound waves to determine the blood pressure of a patient. When integrated with other sensors, the patch can provide a powerful tool for early detection of aggressive progression of COVID-19. Doctors can make informed decisions on when it's necessary to contact a patient, rather than relying on just the patient's self-reports. Next, please. The benefit for customers are threefold. For patients with moderate COVID symptoms, remote monitoring reduces the risk of delayed treatment. For healthcare providers, the device improves efficiency for patient triage and provides a data record if patients do need to come in. For insurance, the device provides potentially a lower overall cost because of the higher efficiency in patient triage. Next, please. Our team is composed of Dr. Sheng Xu, faculty at UCSD and member of NAE Frontiers of Engineering, Dr. Jinghong Li, faculty at UCSD Medical School and pulmonary specialist who brings their clinical expertise caring for COVID-19 patients on a daily basis. Here's myself, who brings device prototyping expertise, and Amar Yagi, who is a serial entrepreneur and brings data security expertise. Next, please. Move forward, we will take an iterative approach for prototype development, and it will be closely tied with clinical validation. By end of the year, we hope to have initial prototype build and start a small-scale clinical trial in Q1 2021. To realize the goal, we're seeking connections to primary care physicians and hospital procurement managers for customer interviews as well as industry connections for clinical and regulatory advice. Next slide. We are covertly monitoring Band-Aids for primary care physicians to efficiently triage COVID-19 patients remotely. Join us and help the medical system allocate healthcare resources more efficiently. Thank you. And with that, I would be happy to take any questions.
Thank you, Sue. Very important challenge here that you are tackling. At this time, I'd like to introduce our ERC panel for this team. I'll be giving a short bio, but fuller bios can be found on the NAU website. So for this round, we have Monty Alger, Professor of Chemical Engineering at Penn State University, Sam Petroda, Chairman of the Petroda Group, Diane Green, Founding CEO of VMware and former CEO of Google Cloud, Dennis Disher, Professor of Chemical and Biomedical Engineering at UPenn, Darlene Solomon, Chief Technology Officer and Senior Vice President of Agilent Technologies, and John Martin, former Chairman and CEO of Gilead Sciences. Reviewers, you now have 8 minutes for Q&A. And the first question will go to Monty. Please take.
Hi, this is Monty Alger. Thank you very much for an interesting presentation. A quick question. Assuming the technology works and you can correlate the data you capture with disease, what's the deployment model? Where would you use this? Populations, universities, cities, hotspots? What's your thinking on that?
Thank you for the great question. So our initial plan is to have the device distributed through the hospital systems on a prescription basis. And it's going to be depending on the doctor's own judgment on who is going to be the higher risk patients, and also only on diagnosed patients.
Great. Our next question goes to Diane. I'm sorry, Darlene.
Thank you. Thank you, Alice, and congratulations, Team Coverly, really nicely done. My question is around the kind of go-to-market requirements, and you outlined 5 vital signs. I'm wondering, where are you in terms of developing the full 5, and what is the minimum set that you think is required to take the path that you just showed for 2021?
All right,
Maybe I can take this question, and you can add on some comments if you have any. So those 5 vital signs actually came from our conversation with Jing Hong, who is also on our team, and unfortunately she has to be on her daily shift today, so she could not attend this event. And those 5 vital signs are tightly related to the practice she is taking right now, and those are closely related to COVID. In terms of the minimum set of symptoms or signals we can acquire, I think those 5 is easily those five sensors are readily approachable with our technique right now, and it can provide enough data for those clinicians to make a informed judgment. So we would say as a starting point, those 5 sensors may be a good choice. And in terms of the status, we have those 5 individual sensors developed in our lab, and as the next step, we can do optimization, refinement, and then integrate them together on a single device that is customized for these COVID patients or suspects. I'm not sure if that answers your question.
Darlene, any other clarification?
Yeah, no, that's great. It wasn't clear that all 5 were this far along, and it's great if you can do the whole set. It seems like it's such an important application that if there are some delays, that the urgency of getting the technology out there would seem very compelling if you do have to make trade-offs. That sounds great.
Yeah, thank you. We have them already individually. Yeah, thank you.
Thank you. Our next question will come from John.
I think John has to unmute himself.
Sorry. Yeah, I'm sorry. Congratulations on the presentation. I agree about the 5 measurements you're taking could be very prognostic. UCSD has collaborations, many collaborations with NIH, and this type of technology could really save the healthcare system a lot of money. And not only that, as we're improving care of Corona or COVID patients, patients are having better outcomes, but there's much more still to be done. And these sensors could reassure patients that they can stay home, they don't have to engage in constantly worrying about getting into the healthcare system because they know they're being monitored in a validated way once you have that validated. So I was wondering if you're engaging collaborations with NIH or other US government agencies to integrate this into their clinical studies.
How about I take this question, Shu? We don't have any collaboration with NIH for clinical validations so far, but the blood pressure sensor was originally funded by NIH. So yeah, so if you have any connections over there that you think would be beneficial for this project, we would be very happy to get introduced and explore any synergies.
Okay. That can be followed up on, I'm sure, right, Alice?
Yes, thank you. Our next question will be from Diane.
Okay, thank you. This looks terrific. As a software person, I'm a little bit curious about how you plan to give people access to this in a way that the hospital will actually adopt it and feel, I noticed you have a security person, so I'm sure you're working hard on patient, protecting patient information. Do you have to integrate this with an EMR system or it's just in the cloud and there's a special interface that the hospital uses when the doctor's looking at it? How do you envision that working?
So our plan is to basically connect with the EHR systems in hospitals and directly integrate the data stream to doctor's office. And we also are aware of regulatory requirements for data processing, data storage, and sharing for patients. And we do have, we brought on a team member who has expertise in data security to ensure HIPAA compliance.
So have you actually spoken with people like Epic or Cerner to see if that's possible?
No, but we have spoken with companies who help to build integration platforms with EHR systems, such as Redox, and basically verify that this idea is feasible.
Okay, great. The software is time consuming, that aspect of things.
These are important questions. Okay, we have time for maybe 1 or 2 more questions. Dennis, you can take the next question.
Can you hear me? Yeah, can you hear me? Yeah, okay, thanks. Yeah, that was great. I was just curious, you seem to say you have all 5 devices already, but not integrated. Is that what I understood in response to an earlier question?
Yes.
Yeah.
You're right.
Yeah. So, what is out there currently commercially that that might be combined with your individual devices? Why reinvent the wheel, so to speak?
Well, let me take this technical question. I think those 5 signals can all be measured with the existing commercial device, but they're bulky. They are constraining to people's daily motion. And they can only measure 1 or at the most 2 to the best of my understanding. I think most of them are only 1 signals they can measure. If you want to combine 5 devices together, that's going to be as large as a desktop. You are carrying a desktop with you all the time, right, to continuously monitor yourself. So our innovation here is to miniaturize all those sensors and only keep those these critical components on board and integrate that with the wireless communication technology, so that is going to be a low form factor wearable device that is customized to these COVID patients or suspects. And that is something that, to the best of our knowledge, has not been available in the market yet.
Thank you, team, and thank you, reviewers. That's all the time we have for questions for this round. I'm going to be introducing the next team, but before I do, we also hope today's conversations will spark new ideas and initiatives. So if you've been inspired, please consider answering the NAE call to action at nae.edu/COVID-19 and enter with your own ideas. The incubator is still going on, and we'll be doing more of these pitch showcases in the following weeks. Maybe I will see you here on stage soon. So I'd like to bring up our next team, handless door opener. Logan, are you ready?
I am ready.
Excellent. You have 5 minutes.
Thanks. So my name is Logan. I'm a co-founder of Handless Door Opener Startup, and our mission is to keep people safer by reducing transmission via surface contamination. Next slide, please. So people already hate touching the restroom door when exiting. In the COVID era, we think people should hate touching all door handles. According to a study, one virus-contaminated door handle can infect 50% of all staff and visitors in a building within just 3 hours. Millions of people assume the risk of touching COVID particles and other germs each day. So we thought, is there a way to avoid contaminating the handle in the first place while retaining function and convenience? Next slide, please. Our solution is a foot-operated hands-free door opener. You can use this device by pushing up on the internal pedal with your toes. This pulls the handle down and unlatches, and then you can swing back with your leg to open the door. We're considering a copper pedal or copper plate on the pedal to ensure self-cleaning. And a quick note on this, a redesign is already underway to improve usability. Next slide. Ideal implementation is a door with any type of latch handle and auto-close functionality. It is easily assembled and mounted to the door by the user, no power tools required. The device completely eliminates the need for hand contact of any kind. And this is our second iteration of the prototype over on the left. The plan is to work with design engineers and optimize further. Next slide. This is not an exhaustive competitive analysis, but a couple of our competitors are shown. The Clorox logo shows constant disinfection, and then the step-in pool over on the left is another competitor. Disinfection is an ongoing cost that requires manpower to buy and use several times a day, but it still retains some risk of transmission. And then step-in pool is great, but does not have the functionality to actually unlatch the door. So its scale and impact is limited to a fraction of our door openers. Next slide. This startup is completely student-owned. We're all engineering students, although some of us have just recently graduated. We were united by the National and Royal Academies of Engineering during the 2019 Global Grand Challenges Summit in London. Our team skill set is comprehensive of essentially every engineering specialty, along with entrepreneurship and patent law. And we've been supplementing business and mechanical design expertise with input from various mentors and other pro bono professional consultations. And we do have sponsorship from Johns Hopkins, as well as we're going through the mentorship program for the NAE. Next slide, please. So we'll launch the product by exploiting our professional networks to connect with target buyers. We plan to implement a try before you buy policy, allowing customers to test out the device on their own building. We also may provide the customers with a first free device to gather testimonials. We do already have a few companies interested in purchasing, mostly in Europe. Archinova is an Italian company in the built environment arena that would allow us to test the product and also potentially become our first customer. And we've been in contact with a few people about licensing with companies like Cintas and Ecolab. Next slide, please. We have a $2,000 investment from winning the Johns Hopkins COVID-19 Virtual Design Challenge. And we've also applied to a couple of accelerators and other grants for additional funding. We plan to have a revenue stream from direct sales to a target audience of small businesses, corporate buildings, universities, labs, and we also plan to have another revenue stream from licensing, like previously mentioned. The profits will be used to send free devices to high-risk spaces like PPE decontamination or healthcare facilities. So doing social good is a very important part of this business. Next slide. Our next steps include prototyping and user testing, strengthening IP protection, and officially launching the business, which includes things like LLC formation, insurance, etc. And we would seek continued mentorship in mechanical design and business administration with an emphasis on international business as we are all from different countries. And next slide. With handless door opener, we can stop the spread, reopen the economy safely, and reduce disinfection costs. Thanks for listening. And with that, I will take questions.
Thank you, Team Handless Door Opener. I'd like to welcome up now our ERC panel for this team. We have Kurt Peterson, co-chair of the Hard Tech Group in the Silicon Valley Band of Angels, that's Silicon Valley's oldest angel investment group. Paul Citron, former vice president of technology policy and academic relations at Medtronic. Neil Payton, former vice president of technology at Helmet Corporation. Roderick Pettigrew, CEO of InHealth, that's a multi-college engineering health initiative between Health Science Center and College of Engineering at Texas A&M. Kershi Tika, she's an independent director on both the boards of Pacific Basin and Ardmore Shipping. Bill Hawkins, former chairman and CEO of Medtronic. And that's our panel for this session. Reviewers, you have 8 minutes. Our first question will go to Kurt.
Well, congratulations on a very elegant and simple and useful device. I'm really excited by this, and I watched the video online. So I'm glad that you have filed for patents on this, filed for a patent, and I really highly encourage you to move that forward. But the question I have is, have you considered partnering with any strategic like door companies or people who build hardware related to doors or installing doors? You know, large companies, these strategic relationships can be very important for small companies like yours. But congratulations again.
Thank you. Yeah, go ahead, Lisa.
Yeah, so we've been in touch with this company, which is the biggest manufacturer of door, elevators, and other systems related to any of these devices. That's the biggest in Europe. And we have contacts with them. So that's a route we wanted to explore after we get the certification, because that's the advice I've been given. So before reaching out to them to have everything certified and tested.
So if I can just add on to that, we want to deploy this first device, and then we want to get into more permanent solutions, which are going to be implemented inside the actual hardware. So that would be more of a long-term goal, but we've definitely been advised to do that, and we would consider that route.
Great, thank you. Okay, our next question is going to come from Bill.
Yeah, congratulations to the team again. You know, you're obviously working on something that is an important problem today with, you know, the transmission of COVID-19. I guess my question is, first of all, from a cost of goods, have you looked at what the ultimate cost would be at scale, and recognizing that if this was to be implemented, you know, it's going to add cost to the existing, building structures? And two, what's the, what percentage of the market is actually doors with latches versus with other means of opening? And have you thought about how big the market is for this?
Yeah, so I've done some pretty extensive financial calculations to the best of my ability, but it's really, really hard to estimate that kind of thing. According to my calculations, we could do about 1% of the market, but the market is an $8 billion market. So I think it would still make an impact. One of the highlighted areas that one of our contacts said was doors for conference rooms in corporate buildings. So we think that could be a big market. But as far as unit cost for the actual product, that's something that's also hard to estimate because we're in the middle of a redesign. So we're thinking about metal versus plastic. We're thinking about adding an up and down track instead of the pedal design. So we're kind of in the middle of completely changing the design. So we don't want to do financial calculations just yet.
Yeah, so to add on to that, like some basic estimates, as Logan said, depend on the building material. So based on making it out of stainless steel, it would be around 40 pounds per device. And this is based on this factory, which is manufacturing a different device. For the plastic one, which can be laser cut, it depends on the fluctuating price of plexiglass, which has been quite high the past month, but it's been lowered now. And that could be around 10, 20 pounds. The quality, of course, then changes with price.
Great, thank you. Our next question will come from Paul.
Yes, thank you. A very nice presentation, and again, I admire the fact that you formed an international team to address this. Have you looked particularly at the school market? And I'm thinking in terms of the children and not the faculty per se, who they don't have the self-discipline necessarily to take measures that they might be carrying with them to deal with the door handles. So have you looked at how a child, let's say 7-year-old, 8-year-old, could use the system successfully in a bathroom situation, for instance?
We are about to start user testing after this current redesign, and we are partnered with Johns Hopkins to use one of their facilities, and we've written a full test protocol that includes a range of ages, shoe sizes, heights, everything like that. So that is definitely something that we're going to be testing extensively before deploying the product.
Good, thank you. Kershi, you have the next question.
Yeah, great job, team, both on the design and your presentation. I guess my question is related to the product design, which I think you already partly answered, but this type of equipment would be used very frequently, and obviously the durability of the equipment is very important. And compared to what your competition has to offer, you have a lot more components. So in your design optimization, are you thinking of maybe simplifying the design so that you could increase the reliability, durability of the design? And you already mentioned that you're looking at different materials.
Mm-hmm. So one of the things in structural integrity was material. We're trying to move towards metal, a lightweight metal, because we think it would just hold up better. And then in terms of the actual modular device that we have now, we think it would be better to move towards almost like a sliding track with the sliding object that you would put your foot on to push up. We think the track design would be, it would hold up better, basically, yeah. So we are thinking about that during the redesign. Yeah, go ahead, Matthew.
Just to add to that, we'd like to try and keep this product as simple as possible, but not only from a usability point of view, but from a manufacturing point of view. So if we sell a product, we'd want to keep maybe the product to have maybe 3 or 4 different pieces so that users, when they're attaching the component to doorways, can do things simply and follow very basic instructions.
Thank you. Our next question goes to Roderick.
I'd also like to congratulate the team for organizing at the recent Grand Challenges Summit and to organize across multiple institutions. That's quite impressive. My major question pertains to usability and the extent to which you've been able to pilot this in the field, what sort of feedback you've gotten from those who have had an opportunity to use it, and the extent to which that has resulted in any design modifications.
Matthew, do you want to take this one?
Yeah, I can take this one. So the initial product that we came up with, we got it relatively quickly prototyped and made. From there, we realized that attaching all the different parts together was difficult, and it wasn't as easy to use from simple materials that can be found in hardware stores. So that means we went and did a redesign. From that second redesign, we then managed to get a few more created and also tested. And the general feedback we've had so far, and hoping to get some more feedback, is that it's not the most stable after a while. The components themselves, the working parts, do work. However, the casing itself, if it was one piece rather than not, would stay attached to the door for longer. And more design, which would be one component for the casing and then one component for the pedal itself. So we're still looking for a lot more feedback.
A quick follow-up. A quick follow-up, Alice, is in your write-up, you indicated that this was applicable to both knobs and latches. How does this function on knobs, and is that still the case?
So there is a separate component which will attach to the door handle or the door knob. We haven't been able to test on many door knobs, as the most of the doorways that we've had accessible to us are handles or just bars that you pull on. So we will have to do a bit more extensive research and looking into it. But we have a basic design which in principle should attach to both a door handle and also a door knob.
Thank you, Matthew. And thank you, team Handless Door Opener. I'd like to introduce our next team up. And again, if you have comments for the NAE or you want to reach out to any of these teams presenting today, please contact the NAE at engcovid19@nae.edu or tweet #NAE COVID-19. You may have ways that you can help these teams that are presenting today. I'd like to now introduce our next team, Team More Efficient and Effective COVID-19 Vaccine Delivery. Are you guys ready to begin?
Yes, I am.
Excellent. You have 5 minutes.
Thank you. Hi, everyone. Thanks for giving this opportunity for us to talk to you about our idea, particularly a novel COVID-19 vaccine delivery using microneedle arrays. My name is Burak Ozdonlar. I'm a professor at Carnegie Mellon, as well as have attended multiple NAE Frontiers of Engineering conferences, joined by Phil Campbell from CMU, as well as Paul Duprex from University of Pittsburgh. We have complementary expertise in medical device design, manufacturing, in vivo experimentation, immunology, vaccine design, and immunogenicity. Next slide, please. So the only way to move forward from our current crisis is to find an effective vaccination strategy. The strategy must include two components. One, a potent vaccine delivery, and two, and importantly, an effective delivery approach. Our current delivery approach using traditional intramuscular delivery with hypodermic needles have a lot of deficiencies. One, it's very inefficient, thereby requires very high doses and causes toxicity, high cost pressures to vaccine manufacturing. It requires cold chain so the viability of vaccines can be preserved, causes needle reuse and needle stick injuries, and it's painful, causing reduced patient compliance. Next slide, please. Actually, an ideal target for vaccine delivery would be the skin. Skin has a very sophisticated immunological network that can trigger rapid and potent immunogenicity, even with the trace amount of vaccine we use in intramuscular injections. However, delivery to skin is very difficult because skin is very thin And current delivery method cannot deliver things reproducibly to the skin, and it also shares some disadvantages with intramuscular delivery. Next slide, please. A very attractive alternative is to use microneedle arrays, which include hundreds of tiny needles in a patch format. We previously designed and developed dissolvable microneedle arrays. These dissolvable microneedle arrays are based on a sugar-based material, and they're also mixed. The vaccine is mixed inside these sugar-based material to form the needles. And after applying this to skin in 10 to 15 minutes, the needles dissolve away and they deliver their cargo into the skin very precisely. Next slide, please. This has tremendous advantages such as dose sparing effect, no pain, no needle stick injury, reproducible and precise delivery, self-application capability, as well as minimal cold chain requirement. However, sterilization is an issue as the material is mixed with the vaccine. We need to use gamma sterilization, which could dramatically reduce viability of live attenuated and protein-based vaccines. FDA approval is also a limitation because anytime we change the bioactive component, new approval is necessary because the whole material needs to be changed. There's a dosage capacity limitation, only dry form biologics can be used, and the dissolvable materials can interfere with the presentation of the vaccine to the immune system. Therefore, rapidly creating a dissolvable eminent platform for clinical vaccination has many challenges. Next slide, please. To address those challenges and retain all advantages of dissolvable MNAs while addressing all of their problems, we came up with a new approach where in this hybrid microneedle arrays, we have stem portion which has through holes from the front side to the back side. In addition to the stem portion, we have these non-dissolvable stem portion. We have dissolvable tips which facilitate clog-free low-force insertion. Thereby, when we apply this patch to the skin, in about a minute, those tips dissolve away and the delivery from the backside through these microcannulas can be achieved. Next slide, please. So here are some application examples. We can flow from backside. We can put the lyophilized vaccine on the reservoir on the back. And we can also add this to existing syringe or create a blister pack to deliver vaccines. Next slide, please. So this brings tremendous advantages. The dose sparing effect, as we talked about, and that causes manufacturing, that reduces manufacturing lead time and cost significantly. It can deliver any type of vaccines, including live attenuated vaccines. It's independent from the vaccine itself. Therefore, the sterilization is separate and FDA approval is simpler. The dissolvable tips can be used to co-deliver adjuvants to increase vaccine efficiency. No special training is required, pain-free, reduces cold chain requirements. Syringes and blister packs can be used, and the manufacturing approach can be scalable. Next slide, please. So what we need to move forward with this approach is two things: research funding to advance the manufacturing technology and validation of delivery characteristics, immune activation, and in humans with COVID-19 vaccines, as well as collaborations with commercial and industrial development to go to GMP production of hybrid microneedle arrays and FDA approval. Thank you for your time.
Thank you, Barak. ERC panel, you are up. For this round of Q&A, we have John, Kershi, Darlene, Monty, Neil, and Paul. And I'm going to give the first question to Darlene.
Okay. Thank you, Barak. And again, really interesting presentation and clearly a next generation of a lot of very powerful research. So at this point, I guess I'm wondering a little bit more about how many of these have actually been produced in terms of the hybrid configuration, and what do you see as the, what is realistic in terms of timing and the most important work to validate the technology?
Very good questions, thank you. We have produced, as you saw in one of the pictures, we have produced a number of prototypes of this. We have, and we can produce in our laboratory tens of these, maybe even hundreds of this a day. What is required right now is actually for us to work with manufacturers to scale up this technology. I have the manufacturing knowledge and expertise to help the manufacturers to implement this technology. In terms of the time scale, if we are aggressive on this and if we actually get the required funding and collaborations, it is feasible potentially to get this into commercial manufacturing within the next 12 months. Now, that's an aggressive timeline, without a doubt, But we are, of course, working towards, for instance, phase 1/2 trials together with Paul Duprex in their phase 1 trial vaccine. So we are also doing things on the background to be able to, shorten that timeline.
Yeah. So it sounds like you're very confident in the validation of the delivery. It's much more about the execution of the scale.
Yeah, because we have a whole decade of experience with the full dissolvable microneedle arrays, and they have been very successful. Now, this one is actually even more advanced. We are, quite confident, but we still have to validate it, as you realize, for both FDA purposes and for acceptance.
Great, thank you.
Thank you.
Thanks.
Paul, would you like to take the next question?
Yeah, sure. First of all, a nice presentation. The question I have is, even in your hollow needle approach, you do have a component that flushes away, if you will, or melts away. Is that going to be a regulatory issue, or is that a material that has already been FDA approved? So I'm actually asking a question that relates to approvability in a short time frame through the agency. And a related question has to do with a non-hollow microneedle, which, from what I understand, shows improved transfer of medicaments through the little holes that they produce. Do a compare and contrast between the hollow needle and the solid needle as far as ease of development and again, the regulatory pathway being as short as possible.
Okay, very good questions. Let me answer the second one first. Indeed, the whole motivation to create this new system was to simplify the regulatory approval, because on the what you call solid needle, which is fully dissolvable, it ends up being a new, in essence, drug every time we mix something else. So that requires a new process for FDA approval. Now, let me go back to your first, and the sterilization is an issue because once we sterilize it together with the drug, we really inactivate the live attenuated vaccines and or proteins significantly, and that's a big concern. Your first question, first of all, our fully dissolvable needles are currently in phase 2 trials, and they have been shown success. The materials we use for the tip, the dissolvable materials, are simple carbohydrates. Okay, these are components like glucose, sucrose, maltodextrin, trehalose, or carboxymethyl cellulose. These are simple and complex sugars. They are by FDA considered as generally regarded as safe materials. We expect the approval for these could be very quick. And not only that, once it's approved, we don't need to go through approval ever again. This system has to be approved once. And once the approval happens, for both the current vaccination and future vaccinations, we can utilize the same system because we are not mixing different drugs into the tip to change, the whole system.
Thank you.
Great, thank you. Our next question goes to Monty.
Hi, thank you very much. Question here on sort of the value proposition here. I think for COVID, I think there will be concern. You always try and minimize risks when you're in a new area. And I can see that with this technique, you have the question of the vaccine delivery, effectiveness, and then this new mode of delivery. Is there a value proposition beyond COVID? Are there other treatments, diabetes, other things that would need this? And what are the advantages of your approach? Is less skilled training? You mentioned the supply chain for cooling, lower dosages. I mean, where can you take this if you miss the COVID window, but you still have a technology? What are your thoughts on that?
A great question. Thank you. First of all, hopefully we are going to catch the COVID window, but this is, as we know, this is not the last pandemic we are going to see, right? No matter what we do, there is going to be future pandemics. Vaccination is a general need. Somebody regard this as, next thing other than water that keeps us alive as human population. So for any vaccination strategies, this indeed, if we go through this development, we'll be able to routinely use this. of course, for things like, indeed, the dissolvable system has a capacity limitation that, for instance, we cannot deliver for, we cannot use for diabetes and so forth, whereas this new system can be used for those purposes. We can deliver many different things. We can do interstitial sampling. So there is a lot of future for this technology. For today's presentation, I focused on vaccine delivery, but we are doing a lot of other things, delivering particles, delivering sensors, doing interstitial sampling with the same technology. And the risks are there, definitely, but I would again perceive the risks to be low simply because we went through more than a decade of experience with the fully dissolvable microneedles. These are even simpler. They are much more direct, accessible to the skin. So for those reasons, I consider the risk to be a medium risk, not a high risk, even though the impact is going to be tremendous.
Thank you.
Okay, thank you. We can take one last quick question from John.
Well, I think your work is terrific. I'm aware of the decade of experience and preliminary data, and I'm very optimistic about what you're doing. Global health organizations all over the world are on parallel to your work, have been trying to get rid of needles. And people are developing vaccines very rapidly for coronavirus. My question is, are you thinking about non-exclusive license so your product, your delivery system ends up with a variety of vaccines, one or two of which may be successful?
I mean, there is definitely a commercial aspect of it, and clearly, because I'm at Carnegie Mellon University, the patent ultimately belongs to Carnegie Mellon University with my name on it. As you realize, we do have with the earlier system, we have multiple patents together with University of Pittsburgh. In this new system, we have a patent by Carnegie Mellon University, a provisional patent that we are applying to the full patent. We still have a little bit of time. In short periods of time during pandemics, a commercial free license could be potentially considered for going forward, of course.
Yeah.
Fantastic, thank you. That's all the time we have for questions. I'd like to welcome up our next team, Team At-Home Diagnostics for COVID-19 infection and immunity.
Hi.
Hi, Jen.
So, hi. I can't see the slide, but I'm assuming that someone is moving my slides. So today we're going to talk about TAP Diagnostics, a rapid point of care test for COVID-19 infection and immunity. Next slide. So what I've shown you here is the Johns Hopkins University COVID-19 dashboard showing that globally there are over 9.4 million total confirmed cases with over 482,000 deaths. And if we looked at the confirmed cases by country, the United States is at the lead. However, we're starting to see under-resourced countries inching up in that leaderboard. And those numbers are only going to increase. Next slide, please. Next. And if we look at the United States unemployment rate, it's at 13.3%, and the projected loss in the US GDP is about 52.8%. Next slide. So the problem, next. Right, so as the US is emerging from lockdown, or parts of the US are emerging from lockdown, the consensus among public health experts is that we need to do regular testing. Next. Next. And we're seeing that even though there are testing centers, there are issues that are arising where there are people waiting up to 13 hours in Arizona. And this is borne out by our customer interviews. Next. So we have patient interviews saying that the most inconvenient part of getting tested is the time. Next. And other interviews where they say, unfortunately, our hospital does not have rapid testing equipment. Next slide. So in terms of COVID-19 testing, the problems are there's 20% false negative rates. It's inaccessible, meaning rapid testing instrumentation are too expensive, especially in low resource settings. And there is essentially no rapid point of care test. Next slide. Our rapid point of care test relies on simple lateral flow assays, just like pregnancy tests. we can get more confident results. And it's reliant on our multivalent proteins that I'll describe later based on our technologies. But for virus and antibody detection, it's more accessible because you can do this at home and then you can do this regularly. There's no need for skilled personnel to perform these laboratory tests. Next. So our solution is a rapid at-home test for COVID-19 infection and immunity. Next. It starts off with a fast finger prick or spit sample. And that's the preferred sampling method indicated by customer discovery. Next. This is then on a paper strip of a lateral flow assay where we can do this in one step and it's easy to use. Next. Which is then reliant on our multivalent engineered protein. So for many years, our lab has been focused on engineering protein self-assembling systems. And we have several patents looking at this, but we've decided to repurpose this and look at trying to bind to COVID-19 virus as well as antibodies. And next. So then this will allow for rapid testing because these multivalent assemblies can also have a colorimetric marker, which can allow testing to be done within minutes. Next. So our diagnostic team starts with me. I'm a professor at NYU Chemical and Biomolecular Engineering Department. And for 15 years, we worked on protein self-assembly and biomaterials. My team is comprised of Dustin Britton. He is our entrepreneurial lead, his expertise is in protein engineering and drug delivery. Farbad Mahmoudinabar, who is a computational protein design expert, and Kamia Punia, our technical lead, who is an expert in diagnostic biomolecular engineering. Next. So from our customer discovery interviews, we identified 3 types of customers: healthcare workers, business owners, and at-risk low-income community residents. And from these interviews, we understood that they want higher accuracy, less retesting, more comfortable testing, confidence in employee safety for business owners, and cost-effective testing, as well as easier access to tests. Next. So our value proposition are threefold. One is the multivalency in our design improves sensitivity, creating confident results. Low cost and simple design allows anyone, anywhere access to use these at home. And then individuals could regularly test so that we can resume normal activities. Next. We identified 2 beachhead markets, local stores and low-income communities where current testing is cost limiting, and on-site critical businesses who want to provide regular testing for employees. Next. So we have the next steps for the couple of months that lay ahead of us in terms of technology and business. Next. So the first couple, next, we're gonna generate proteins, optimize the proteins in terms of technology, and then on the business end, focus customer discovery with businesses and low-income communities, and then assess the value by testing. Next, we're gonna test protein binding affinities. Next, and immobilize them on paper strips and begin scale up. And then on the business end, develop business model and apply for FDA approval. Next slide. What we need from you. Any type of collaboration with COVID-19 testing laboratories, connecting us with people with scale-up expertise, connecting us with someone to help navigate the FDA approval process, as well as confirm feasibility of the adoption process with business owners. Next. So you can contact us with me and also my entrepreneurial lead, Dustin Britton, and I'm happy to answer questions.
Thank you, Jen. And again, our audience at home, if you have ways you can help this team or comments or thoughts, we encourage you to tweet hashtag NAE COVID-19 or email engcovid19@nae.edu. Review panel, you guys ready?
Yes.
Excellent. For this round of Q&A, we have Paul, Bill, Roderick, Dennis, Sam, and Diane. And our first question will go to Roderick.
Oh, thanks. Wonderful project. I gotta say, I really like this project. As my many of my colleagues at the NIH, specifically at NIBIB, will know I've been advocating for just this type of thing for at least a half a decade. So hats off to you. And I agree with your premise. I agree with your statements about the impact it would have and the good it will do. The questions I have are a little bit of a technical nature. What is your mechanism of readout? Is it based on color? Is it based on fluorescence? And related to that, with the samples that you plan on using, blood and saliva, what do you know what your detection limit will be in terms of the number of virus particles, the minimum number needed?
Right, those are excellent questions. So for the question about detection limit, we are in the process of actually generating these proteins. So we have not yet determined that. But based on our research and studies from other systems for looking at self-assembling systems, we're pretty confident that we'll get pretty high detection, but I cannot quote the detection limit. And then, sorry, for your first question, it was on
your readout. How do you
readout?
What's your
so yeah,
signal?
so the readout is colorimetric. So we have two designs for the colorimetric readout. Just gold particles as one, and then another is just actually a colored small molecule. And so those are in our designs. Thank you.
Okay, thank you. Our next question will go to Bill.
Yeah, this is awesome. I mean, clearly a huge unmet need here. The question I have though is, I mean, I know this is more of a concept based on this protein technology, but you know, one of the challenges now that we have with the current testing, as you pointed out, is the level of false negatives and even some, you know, false positives. Why do you think, what is it that you think is unique about what you're doing that could, you know, help us to address that problem and develop a test that would, you know, improve upon the false negative rate and even perhaps, you know, have, you know, the level of false positives that would be acceptable for clinical practice or for routine practice?
Excellent question. Yeah, so our technology is based on having multivalent display of protein targets so that you can better undergo protein-protein interactions. And our hypothesis is that with that multivalency, we'll get higher sensitivity. And so by doing, our goal is that will allow us to get higher sensitivity for the antibody, because we're doing, in our case, 2 tests in once, right? So we're doing the virus test as well as the antibody test. And in both cases, actually, in either case, they have different false negative rates. But all our technology relies on protein-protein interactions, and we hypothesize that having this multivalency will allow for higher sensitivity, especially with antibody, and lowering the false negative, and then with the virus itself. And so that's what we believe. But we're planning to test it all out, and we're in the works of doing that right now.
Excellent. Dennis, would you like to take the next question?
Yeah, again, that was really terrific, thought-provoking. I was kind of curious for capturing the virus with ACE2. I guess there's two questions I have there. Is there's other factors, other coronaviruses that might bind this?
Right.
Specificity, but why not use an antibody? It's already bivalent, maybe an IgM, which is higher valency as well against the antibody. What the coronavirus affinity for ACE2 is monomeric. Is not as high as it is for an antibody, but just the technical details of how to detect the antibody, the virus.
Sure. So you're right. One can use an antibody to detect the virus, but what we're hoping on or relying on is that we have multiple display of these proteins, so that hopefully just the ability of having this multiple display will allow for any small, tiny amounts of virus protein to be able to be detected onto our multivalent display. And so, yes, those are technologies that currently exist, but we're hoping that what we're going to be providing is higher sensitivity by virtue of this multivalency. Now, we can do this and test it out, but we still need to kind of run these experiments, and we have lined up an expert who's actually doing blood sampling and biological fluid sampling for us so that we can actually test on real samples, because I think that's where we can get to understanding sensitivity and how well our system will work.
This is great dialogue. We have time for maybe one or two more questions. Diane, you can take the next question.
Oh, thank you. So this is pretty exciting. Just a few questions. Like, I'm curious when you would have the testing done. Maybe you said that and I missed it. I was curious what you mean by help in scaling up. And then just kind of a fun idea I had immediately was if you could integrate with the contact tracing apps, just take a picture of this when it detect, the color, and have it automatically go to the contact tracing app. That would be a nice way to do vertical.
Yeah, those are great ideas. So in terms of the first question, in terms of timeline, we're constructing them in the lab currently at NYU, and so we believe that within 3 months we'll have our prototype, and we would like to test that out. In terms of scale up, maybe that is this might require some iterative cycles where we're doing this more than once. But if we're very optimistic, then we can go from there and then do scale up from there henceforth. Sorry. And then in terms of your suggestion about linking it to contact tracing, that would be what we would want to do. That's and also we were also thinking about how are we going to get these results to doctors or get these results to the healthcare practitioners, because it has to be gone through a healthcare system. So I think that is an excellent way to do that as well.
Excellent. Our last question comes from Sam.
I just want to know how much would it cost per test?
Right, that's an excellent question. And currently we have not done any assessment of the cost, as most of what we've been focusing on is trying to generate the construct so that we can lay it on the LFA and do the assessment. That is our next steps, I guess, for our team to figure that out and sort that, what the cost would be.
Any guesstimate?
Any guesstimate. Guys on my team, do you have any guesstimates?
I don't see why it would cost more than a pregnancy level LFA, but I think what's more important is assessing the cost based on who we're directing it towards, which would be like small businesses or low resource areas. So I think that's what's more important in deciding that price, but we still have to assess it.
Okay, thank you, team At Home Diagnostics for COVID-19 infection immunity, and thank you, reviewers. I'd like to introduce our last but not least, certainly, team, Self-disinfecting N95 masks.
Hello, my name is Melissa Banks, and I am the head of ARIS, where we research and design copper coatings for N95 respirators. First off, I think I can safely speak for all of us here when I say that 6 months ago, none of us dreamed we'd be here today troubleshooting a pandemic. And yet here we are, because problem solving is what we do and is how we fight back. But there's another group of professionals fighting back that do not have the luxury of doing it via lab or from across Zoom, and those are our frontline medical workers. COVID has not been kind to anyone, but these are just a handful of the medical personnel we've lost to this virus. This is Celia Marcos, On April 3rd, clad only in scrubs and a surgical mask, she ran to the aid of a COVID patient who just 2 hours prior had asked to go home because he, quote, didn't feel sick. By the time those 2 hours had passed, his heart had stopped, and she arrived to perform life-saving chest compressions despite being without a proper respirator. 14 days later, and Celia Marcos was pronounced dead as a result of COVID-19 infection. The reason Ms. Marcos wasn't wearing the recommended N95 was because they were being kept under lock and key at her hospital. Global supply shortages have forced many healthcare facilities to ration or reuse these masks, often with grave consequences. So this is the problem we have endeavored to solve. There are 3 primary obstacles to resolving the risks and supply shortages associated with N95s. First is that there's no proven method of disinfection. The second is prior to the COVID pandemic, the CDC recommended disposing of N95s after each encounter with an infectious patient to minimize the risk of transferring a bug from one room to another. Third is contamination risk. The longer you wear an N95 respirator, the more active virus you accumulate directly above your nose and mouth, which is where you're most susceptible to infection. All 3 of these issues could be solved if we could develop a self-disinfecting respirator. So why copper? Since 3200 BC, mankind has used copper as a disinfectant. Unlike other heavy metals, it has a single unpaired valence electron, and that electron binds with oxygen from the air to form a highly reactive ion orbiting each atom. When a virus lands on a copper surface, the ions shoot through its membrane like tiny subatomic bullets, leaving behind fragments that are then incapable of infecting. The end result of this process is a self-disinfecting surface. And on the right-hand side, you can get a better idea of the antimicrobial properties of copper in comparison to silver ions, for example, or stainless steel, which is a very popular material in hospitals these days. So what we propose is making this process work for us by combining the antiviral properties of copper with the pre-existing filtration within N95s and applying it as a coating to the polypropylene fibers that make up the respirator. When I first got the NAE's call to action and read through it, my first thought was, how can I help? But my second thought was, how can I help fast? So I didn't want to reinvent the wheel. I wanted to design something that could take a good that was already in production and alter it in a way that could dramatically extend its shelf life. The most sophisticated respirators in the world aren't going to do us any good if we can't make them quickly and we can't make them affordably. Pre-pandemic, hospitals paid roughly $12.18 per employee per week in masks. This doesn't account for hazardous waste disposal costs or storage of the large quantity that's required to meet hospital demand. It also doesn't factor in that COVID-19 can live on a typical N95 for 3 days. Copper kills COVID in under 4 hours. And I want to make something clear, it never stops working. So you may have seen that pretty patina that coats most copper surfaces over time. Those surfaces are just as antimicrobial as the shiniest pots and pans inside of Williams-Sonoma. Oxidation and the passage of time do not affect this attribute. So the shelf life of a copper-coated N95 is only limited by the underlying material's ability to maintain a proper fit, potentially months on end. But I've used 1 week as a conservative estimate that I'm fully confident we can exceed. A 1 week time use would make the price to beat $12.18, which is exponentially more than the cost of materials and coating via the mechanisms we have mapped out. Finally, although it may not seem like it, a pandemic by its very nature is a finite period of time. And if I was going to invest time and energy into this project, I wanted it to be something that would have applications beyond the situation we find ourselves in right now. Widespread use of copper coatings could revolutionize everything from decreasing hospital-acquired infections. Sorry, slide. Oh, okay, there we go, thank you. To cell phone covers, keyboards, grocery cart handles. It can help us fight against antibiotic-resistant bacteria. It has uses in non-healing ulcerations and disrupting biofilm. So the future is very, very bright for this material and for this technology. So thank you. Thank you for your time and your attention. And as much as I've enjoyed introducing this project to all of you, what I'd love to do now is just pipe down and grab my notepad and take down all of the advice and comments and critiques you have for us, as well as answer any questions alongside my partner, the lovely, talented Miss Samiksha Agrawal.
Thank you so much, Melissa. For this round of Q&A, we have Bill, Dennis, Rod, Neil, Kershi, and Kurt. And I'm going to give the first question to Bill.
Oops. Yeah, really terrific presentation. I guess, I think you're going on the right path on looking at the cost-benefit analysis. Obviously, with the struggles that hospitals have today, just on their basic economics. I mean, taking on, additional cost. I mean, notwithstanding the benefit here is going to be, it needs to be cost effective. One thing, just as an FYI, I'm involved at Duke, and we have found a way to decontaminate masks using hydrogen peroxide vapor. That's just something that you should know about. I mean, it's got its issues. I mean, you can extend the life of a mask by using hydrogen peroxide. But look, I think there's a big unmet need here. I like the idea. I think the question is going to be, I mean, can you make it affordable?
Is that a question for me now, or?
Yeah, I mean, I guess the question is just what the, I mean, how much cost do you think it would add to a mask?
With the primary material that I'm interested in using is cuprous oxide. So there's the nanospheres about 480 nanometers, and a coating would probably run an additional $1.25 per mask.
Mm-hmm. And that includes the cost of putting it into the mask, not just the material cost, but the assembly.
Correct. Yes. So it would be it would be attached through it would be produced through solution modification and then attached with a silane coupling agent.
Yeah.
And the
Great,
thing
thank
is,
you.
sorry. And the thing is that it would really, the thing is it would greatly extend the life of the mask as well. So it would, in hindsight, it would actually end up decreasing the need for multiple masks and therefore reduce the need for and the price automatically due to supply and demand in that way.
Great, thank you. Our next question will go to Neil.
Yeah, I'm concerned. I guess, am I unmuted? Yes, I am. I'm concerned about the long-term effectiveness. As you know, copper oxidizes very rapidly, and you're saying that copper oxide is just as effective, but I think that needs to be proven. And maybe you have to your satisfaction, but not to mine. So that's my real question is, what's the long-term effectiveness of this treatment?
So copper oxide has been tested as a antiviral coating in long-term studies. It's actually shown to be more effective than pure copper. And additionally, the nano, if you're able to use a nanosphere coating, it's the grab, I guess you'd say the tooth of the nanospheres can more effectively grab the virus than like a smooth metal kind of coating. But the oxidation has been studied, just like they use copper and brass in railings and handles from some of our city halls that were built, 100 years ago. They've tested, and those are still just as antimicrobial as they were when they were first installed.
Okay, thank you.
Okay, Kershi, would you like to take the next question?
Sure. So I've seen some. By the way, a great presentation. I've seen some ads on copper masks, and I was just wondering. I haven't really studied them, but I was wondering what's different. I guess they may not be N95 masks. Can you just comment on that?
Yeah. So right now, a lot of people are trying to just harness the power of copper and use it in masks. There's fabrics that have it woven into it. The main difference between a mask and a respirator are the fit. So a mask is going to be anything that just attaches behind the ears and is kind of gapy here. An N95 respirator can say that it cleans or filters 95% of the contaminants because it's filtering everything that you're breathing in. So it has a seal that goes around your face here. So the masks that you see, like surgical mask type setups, those aren't going to be N95 or rated that way, just because they can pull in air from the pockets on the side.
Roderick, did you have a question?
Yes, actually, my question was somewhat related to Neil's question, but first let me congratulate you for the concept of the self-disinfecting mask. It's a great idea. But I question as to whether or not you know what the level of copper that is needed in order to have this biocidal effect, specifically for the coronavirus. Is the effectiveness of copper and copper oxide, are those equivalent? It's sort of related to Neil's question. And have you looked into any potential toxicity related to copper nanoparticles that might be in the mask and might be inhaled?
That's a great question. And the nanoparticle aspect is still one that we're looking at, whether we should be using bulk particles or whether we should try to go down the road of nanoparticles, just because the engineering is newer. And so the long-term health implications are less certain. Copper as a material in itself has been used in implantable devices, pacemaker portions, intrauterine devices since 1967, I believe. So it is biocompatible in that sense. But again, with the nanoparticles, that does require additional testing to make sure that the adherence is sufficient to not cause any respiratory distress.
Thank you. And thank you. That's all the time we have for questions. Thank you, team Self-Disinfecting N95 Mask, and thank you to all of our teams. This concludes the pitch showcase portion of our program today. Please join me in giving a virtual round of applause to these impressive teams and technology showcased. Each has made spectacular progress in a very short period of time. These are truly heroic engineers. I'll now like to invite Paul Citron, Chair of the Expert Review Committee, to share some closing remarks on the presentations today.
Thank you, Alice. Engineers have it in their DNA to make things better. They apply their ingenuity to essentially all aspects of life. Sometimes they make breakthrough innovations. More often, they make useful and valuable advancements that build on earlier innovations. It's what engineers do. When solutions are needed, engineers are there either as a starring role or in a supporting role as enablers. The sudden emergence of COVID-19 was and is a game changer. It poses a serious threat, sometimes deadly, to those who contract the virus. COVID-19 has overwhelmed hospitals around the world, exhausting caregivers and unfortunately infecting many of them. Its impact cuts across many other aspects of society: social interactions, travel, the economy, jobs, education, supply chain integrity, disaster resilience, and patient access to non-healthcare issues, the so-called elective surgeries, for instance, and the list goes on and on. The wide impact of COVID-19 has spurred initiatives to develop more effective approaches for detection, treatment, mitigation, and ultimately prevention of the disease. The NAE's call to action against COVID-19 is intended to harness the creativity of engineers, whether they are still students in academia, industry, or retired. It is a multi-generational effort. The 5 projects pitched today each potentially address an unmet need in the battle against the pandemic. Please note that today's session is not a culminating event. Rather, it is the first in a series, and our idea incubator is still ongoing. I'd like to provide my summary observations regarding today's presentations. Team 1. The current pandemic has accelerated the need for wearable monitoring systems that are sufficiently comprehensive to assess the patient's condition and alert if the diagnostic trends require action by medical personnel. System accuracy and ease of use by the patient and caregivers are very important. In my opinion, device cost is less of a concern if the array demonstrates high overall value. So value is important to assess. Inclusion of a robust wearable continuous blood pressure sensor should be of keen interest and has uses well beyond COVID-19. It's sort of the holy grail. Team 2, handless door opener. First, it is refreshing to see an international team of students address a serious source of infection. Yes, there are many automatic door opener systems, but cost and complexity of retrofitting get in the way of application to the installed base of doors. This team's approach may provide a practicable solution, especially for interior school doors, where children may not have the self-discipline to take other protective measures when opening and closing doors. Team 3, the more efficient and effective COVID-19 vaccine delivery. The skin is a very complex organ. This group has recognized the role of skin as a possible vaccine site through the use of hollow microneedles. If shown to be effective for a COVID-19 vaccine or for other medicaments, such an approach would be a simply elegant delivery modality and could help make vaccines more widely available. Team 4, point of care, at-home diagnostics for COVID-19 infection and immunity. Potential reliability, cost to manufacture, and simplicity of use are potential hallmarks of this innovation. If available, it would have a positive impact on infection screening and provide an instrument for containing infection spread, and also permit safer work and other public space environments. And finally, Team 5, copper-coated reusable N95 respirator. This is a clever concept and addresses the issue of an environmentally contaminated mask becoming a source of infection to the wearer. It also has the potential of reducing mask supply issues because of safer reuse. An important issue is the intellectual property landscape, as well as securing interest from one or more mask manufacturers. For those in the audience who have inputs regarding any of the pitches or would like to help this initiative in any way, please contact the NAE at engcovid19@nae.edu. If you would like to enter the call to action incubator with ideas, please go to nae.edu/COVID19. I'd like to thank today's teams for responding to the challenge and proposing a piece of the puzzle to mitigate the effects of COVID-19. My thanks to the Expert Review Committee for their insights, Alice Liu for moderating, and also to the NAE staff, including Randy Atkins, Brandon Green, and others who have helped put this endeavor together. We hope to host you again in future pitch rounds. And with that, I wish you good health and a good weekend. Thank you very much.